Here, the little vessels, if we take a cross-section of those vessels, this is what it looks like. So these little circles are the red blood cells. Here's the wall of the vessel. And then in this sort of lighter shaded pink here is the smooth muscle cell wall, okay? It's the wall of the vessel. And the smooth muscle cells are very important for keeping the vessel at the right tension. So when you get high blood pressure, your cerebral blood vessels relax because it wants to make sure that it's appropriately responding to the change in pressure, and the smooth muscle cells are critical to that response to the change in blood pressure. And the smooth muscle cells are sitting here. These are in blue, so they are surrounding the vessel, and the key protein we're going to talk about today is Notch3, and Notch3 is sitting there at the membrane of the smooth muscle cell, okay? So it turns out that mutations, so changes that are inherited in the Notch3 gene, is what causes Cadasil. So why is that? We don't know. Let me just stick with that. These are supposed to come in order so that you didn't get too busy, but it's too busy. So we'll just go, let me just take you through. So this is the Notch3 protein. It's sitting here on the membrane of the smooth muscle cell within the vessel wall. And it's a big protein that interacts with this environment. When there are changes that are associated with Cadasil, what happens is the pattern of the little protein components change. There should be an even number of one of these protein components called cysteine. When there's a mutation that causes Cadasil, you get an odd number. That's just one mechanism by which notch mutations affect the protein. So it affects the structure of the protein. Now normally, when Notch3 is sitting there, it functions to take signals from the external environment and transduce it to the intracellular space. So the vascular smooth muscle cell can then respond to clues from the outside. And this protein is critical to doing that. So it gets a signal from the environment. And part of that signaling is that it gets cleaved by an enzyme right at the junction of the membrane. And then this intracellular part now moves through the cell, and now that it's moving, it can then initiate signaling to start other functions going on in the cell, okay? So that's one important part of Notch3. So one problem could be that when you have mutations in this Notch3 protein, you get a change in its normal function. There's something wrong with the way it responds to its environment. There's something wrong in which it lays itself ready to be cleaved, and you can impair the function of Notch3. So one possibility is Cadasil is caused by a lack of normal function of Notch3 because of mutation. But another thing that we've learned, and I'm going to show you a little bit of pathology in a few seconds, is that when we look at the skin, if we look at vessel pathology from patients that have Cadasil, there's abnormalities in the vessel wall. There's also accumulation of protein. Very odd, unusual, and shouldn't be there kind of protein collections. And one of the peptides that are near those protein collections is Notch3. So when this protein gets cleaved, it releases this intracellular part, but it also releases this extracellular part. And this kind of goes floating off. But when there is a patient with Cadasil, you get an abnormal accumulation of this protein, which then sits there. And another hypothesis is that too much accumulation of this Notch3 protein also causes damage to the cells in the vessel. Yeah.
Is this in a living human being, or is this a post-mortem autopsy?
That's a great question, because we have information on both. So in both of those cases, you can see the abnormal accumulation of the Notch3 extracellular protein in those protein collections. Okay, so all this to say that Notch3 mutations causing Cadasil can be a loss of its normal function, or it could be because there's a toxic accumulation of something that shouldn't be there. We don't know yet. Also of note that I want to point out here is the work that we're doing to understand the function of Notch3 and Cadasil is very important, obviously, to develop therapies for Cadasil, but it also turns out to have huge implications for how we potentially manage small vessel disease in general. So this is a type of stroke that affects at least 25 to 50% of stroke victims. And what I want to show you here are a couple things. That first of all, it's frequent, small vessel disease. Second, that it's a major cause of stroke and dementia. And third, that at this time there's no real good management. In fact, the stroke trials have been probably as successful as Alzheimer's trials, which are not successful. So they have a lot of interest in knowing what we can do from the genetic modeling perspective to learn more about small vessel disease, and they consider Cadasil as a potential small vessel model. And I just want to show you just to demonstrate the similarities, because the same type of vessels are involved in both diseases. You can see similar clinical manifestations, pathological manifestations, and here at brain MRI, I couldn't distinguish between the two. And here's an example of just how these are similar even in a pathological sense. So here's a normal brain, an aged normal brain, and the brown is standing for a smooth muscle cell protein. So it's giving you an idea about how intact those smooth muscle cells are within the vessel. Here's someone that has typical small vessel disease with high blood pressure, diabetes, etc. There's an abnormality. They do not have a coherent strip of smooth muscle cells. There's loss of smooth muscle cells. And similarly in Cadasil, there's loss of smooth muscle cells. So this led to a study which I'll talk about by an investigator at Harvard just a couple of years ago, who was able to show that if you can somehow boost signaling to Notch, you can actually rescue some of this loss of smooth muscle cells. So that's a really interesting point. So this is a biologically, clinically relevant readout of dysfunction, the loss of smooth muscle cells in the vessel, and by increasing Notch signaling, you can rescue that. Okay, so what are the next steps? What does all this mean? I think that was all just background to say, number one, if we want to create drugs, we need to know something very important. How do we design the drug to the right molecule or pathway in the cell? And what I've already sort of talked about is there's evidence to support both, either a toxic accumulation or a loss of function. And that, again, studies that have enhanced Notch3 signaling can actually improve the smooth muscle cell vitality. So what do we need to know? We need to know how do all Cadasil-causing Notch3 mutations impact the function and the binding of the protein to other proteins. Number two, we want to explore the utility of modulating Notch3 function in other small vessel diseases. So if this works in Cadasil, is it going to be applicable more broadly? Can we help other people? And so for that, we need to know, and again, because they share so many features, and for that, we need to identify people with small vessel disease who have abnormal Notch protein profiles, and then they might be the right ones that you give them the Notch medicine to. So this is sort of like a towards precision medicine thing. Can you take someone in the clinic that has a stroke, take a little example of their skin, and then assay their sample and find out if they act like they would respond well to Notch3 boosting. So what we need to know is, what is the spectrum of Notch3 function among people of all types of stroke diseases? And then lastly, we need to know what regions of the protein are particularly important, right? Is it all or not? So we need a systematic investigation of each small building block of the protein, which can then tell us which region is particularly important in causing disease. So we can screen individuals who may have also small vessel disease and then more intelligently interpret their Notch3 variant. They look like they have Cadasil. Let's take a look at how their Notch3 is functioning. This really seems like it's Cadasil. And that's important because it's important for therapies, but it's also important because they've got to tell their kids if their kids are at 50-50 risk of inheriting something that can manifest in stroke. So we need to know what are the functional consequences of Notch3 changes at every position across the protein.
How many amino acids in the Notch3 normal protein?
Yes, many thousands.
Amino acids, it's like 1,700.
700.
So that is sort of the general framework, and now I'm going to hand it over to Adam Gordon, who's going to be able to put even further framework on this idea of what can genomics.
Before you go, just one question. Do you think that there's a proportion of undiagnosed Cadasil patients
Yes.
that end up being just stroke?
Absolutely, and I don't know, was that a question?
Yeah,
Adam's going to talk about that.
So thanks, Sumi. Did it go? Yep, okay. So my name's Adam. I'm a postdoc over at UW Medical Genetics with Gail Jarvik, who's my PI and the head of the division. And I want to take a step back first and talk a little bit about some of the work that has been done nationally in genome sequencing, how that relates to disease and also in healthy people. Generally understanding our genomes and the variants they contain. So you may have seen this slide before. It's somewhat of a cliche among geneticists by now. But basically, over the past 10 to 15 years, the cost to sequence an individual gene or genome, by sequence I mean read it, to read out every single letter of your genome, it has fallen dramatically. And it's almost at about $1,000 now to read an entire genome. Alongside this decreasing cost, it won't be long until you and your doctor will be ordering your genome. You've probably heard of these two companies. This is 23andMe on the left and Color Genomics on the right. They're under $300 each of these, and you can get your genome sequenced or a large portion of your genome typed by these technologies right now. In addition to that, there are large programs out there called All of Us, which are aiming to sequence the whole genomes of millions of individuals across the entire country over the next 5 years. So we're about to receive an avalanche of genomes, and many more people are going to have access to their personal genomes who didn't before. So what actually is in a genome? And I should mention, please feel free to interrupt me at any point or raise your hand if you have questions. Genomes are complicated. So a genome is it's just about 3 billion letters. So it's made up of A, T, C, and G, and if you open a genome in a text file, it's literally just 3 billion letters sitting there. Hard to make any sense of. Of these 3 billion letters, only about 1% of them are in genes, which are the recipes that encode proteins. And that 1%, that tiny piece of the genome, is what we call the exome, which comes from exons, the bits of genes that make the protein. So since 2009, when the first exome was sequenced, we as a field have sequenced tons of exomes. We're currently at hundreds of thousands. If you want to play around with some of this data, a lot of it is publicly accessible. There's about 140,000 of them at this website here, EXAC, which is the Exome Aggregation Consortium. You'll hear that come up again. So I joined the Department of Genome Sciences as a graduate student in 2009 because I was interested in this rapid expansion of sequencing and understanding what at the time was the first exomes that were first coming online here at UW as part of the NHLBI exome sequencing project. So through that work and through the work of others working on exomes, we learned that that each of us has about 100 to 400 variants within our exomes that change the actual sequence of the proteins. We call these missense variants. So if one amino acid building block for a protein was, say, alanine, and in me, and the lysine in you, that would be a missense variant. So depending on what your ancestral background is, each person in here might have 100 to 400 variants that do change the sequence of your protein. And we don't really know among all of these how many of them are actually likely contributing to disease or phenotypes or traits. Yes?
Sequence could mean the order in which
Right.
genes appear. Is that what you're talking about here?
Kind of. It does.
I mean, are genes going to be moved around?
In some people, they are. In most people, though, so our
Are you
genomes
talking about a one codon change in these variants?
Yeah. So the codons are the piece of DNA that actually makes an amino acid, and so it's one base change in that codon, which will then turn it into an amino acid.
Okay, could I restate your point here then? Is that each of us have 1 to 400 single codon changes across our entire exome?
Yeah, more or less. Yeah, we call those missets, though, right?
I'm a software guy, so I don't know jargon. Okay, thanks.
Yeah, but that's correct. Yeah, the DNA is it's 3 letters for 1 letter of protein language, basically, and then when we call a change in that protein language from one amino acid to another, that's the jargon, the missets variant. So we all have a bunch of those. And we know that some of them cause disease in some people, but all of them don't cause disease in all of us. So there's got to be some kind of subset. We don't actually know, though. So about 50 of these in any given person have been associated with disease by someone at some point. That includes all of us. So most of these probably aren't actually causative of serious disease, although some of them are. In addition, if you just look at what's called damaging by there are a bunch of computational algorithms out there that claim to be able to sort these variants into the ones that cause disease and the ones that don't. They don't do a great job right now looking across your entire genome. And of all the variants that you have in your genome, 75% of them would be called damaging by the current algorithms. So we need a better job sorting the ones that cause disease from the ones that don't. So that leads to my current work. I've been working on a project called eMERGE, which is funded by the NHGRI, the National Human Genome Research Institute. eMERGE stands for the Electronic Medical Records and Genomics Network. And basically the idea of this network is to collect patients from all these various health centers across the country, have the patient's genomes sequenced, or here it's actually a portion of the genome, the exome, link that exome directly back to the patient record, depositing it back into the electronic health record, and then aggregating this data and the experiences that the patients have and their clinical outcomes across all of these sites in order to better sort some of these variants. So to date, we've sequenced about just under 16,000 people in eMERGE. And again, because these are patients linked to electronic health records, we can go and follow up about the variants that actually cause disease and look across all of these different settings. So if you these are people who are unselected for disease or family history. So basically sort of grabbing more or less people off the street, people walking into a health center. In general, what we find is that 3.5% are going to be positive for what we call an actionable variant. So this is a really small set of variants that's literally, we know they cause disease, but not only that, we know that we can do something about them. They cause disease, and also they indicate an action, a clinical action that can be taken, that should be taken for that patient's benefit. So actually 3.5% of healthy people are going to have a result like this, just coming in off the street, which is a large number of people as we start getting up into the millions, etc. So looking at just the types of stuff you're going to find, various forms of hereditary cancer risk are going to be the most common. That second one there is another cancer risk that's separated out because it happens to be more common. That APC guy there. And then you'll have cardiomyopathy or heart disease and long QT, which are arrhythmias. And then you get things into like high cholesterol and hemochromatosis and iron metabolism. And then there's a whole bunch of stuff, including Cadasil in this other category down here. But we're going to find
BRCA.
Sorry?
No BRCA.
BRCA is included in cancer.
Yeah, cancer. Fine.
Yeah. So again, we're going to find a wide range of stuff from across the spectrum, and these are just, remember, the variants that we know cause disease. But there are a lot of variants that come up in these genes that don't cause disease. So when we read someone's genome and we find all of the variants, how do we actually sort all these missense variants that come up into these are the ones we know cause disease and these are the ones who don't? Well, there are a set of guidelines, so basically a set of rules that was come up by the American College of Medical Genetics to classify these variants. And they end up in these following categories. There's 5 of them. So first, pathogenic or likely pathogenic almost certainly causes disease. It's hard to get into this category. You have to have a lot of evidence supporting you. I'll show you the rubric in a second. And then on the other side, you have benign or likely benign. We've seen this in enough time in enough people that we know it doesn't cause disease, or we know it doesn't affect the protein just because of by nature of what it does to the actual sequence. And then there's the ones that keep you up at night, the guys in the middle. So these, you may have heard this term before, they're called variants of uncertain significance for the US. And these are the guys that literally just could go either way. We don't know. We don't have enough evidence. Either it's the first time we've ever seen them before, or we've seen them in a few people, but not enough to know whether or not it's causing disease or not. So again, we're still left with all these variants in the middle in which we need more data right now to understand.
Adam?
Yes?
Can you differentiate when you're looking at these VUSs between like a single missense versus a collection of changes in a gene that when together are enabled that then they cause together a particular disease?
Yeah, so because this disease is pretty rare, most of them are actually a single mutation, caused by a single mutation. Often the rarer and more severe genetic disorder is, the fewer variants it's caused by. So the things that are Mendelian and by that I mean they pass directly, following inheritance rules perfectly. Those tend to be caused by single events instead of.
And how do we know that? Do we know that just from a whole body of data?
Looking at individual families that have this disorder and painstakingly sequencing individual gene by individual gene before we got to this technology and identifying, oh, this gene has a variant, this gene has a variant, and then going and testing all the other family members, the ones who are affected and the ones who aren't. we can basically say whether we're sure or not we got the right one,
Mhm.
right? That's actually, that process is called linkage and segregation. And it's really important, actually, a good segue, because these are all the rules, basically. These are all the criteria, and you're not designed to read this other than to say it's complicated. But these are all individual criteria you can assign to a certain variant based on information you have at hand or information you can go to generate. And there's an entirely other set of rules about how each of these criteria add up to fall into one of those three categories at the end. It's like a point system. You have one of these points minus, you have one of these criteria minus one of these plus 2, etc. So I wanted to highlight a couple of these. So this one, this guy here, which counts as a supporting criteria, is multiple lines of computational evidence support a deleterious effect on the gene. So again, and these are pretty general. So for a lot of variants, we don't have multiple lines of computational evidence. We might have one line of computational evidence or disagreeing lines of computational evidence. But we don't have great computational predictors yet that allow us to use this criteria with regularity across the entire genome. Also, a much stronger criteria, but is hardly ever used, is well-established functional studies show a deleterious effect. It's been really hard historically to go through and measure the function of individual genes one at a time, and even harder to do so for every single allele that we know might cause disease or suspect causes disease. And then finally, to James's point, so segregation data, there's literally just an arrow. It's been since somewhat better quantified, but we hardly ever have access to this kind of data, which requires participation of families and engagement of families, patients, and their doctors.
In a
Yes.
future where we had 100 million genomes sequenced completely, and complete health data and performance on those 100 million individuals, you just do a big, massive machine learning thing, and this whole thing would probably disappear. Does that sound
I
correct?
would think so, but the problem is there's so much variability among individual diseases, and for example, certain diseases might be caused by, we know it's a loss of function of the gene. We know that when you give when you put that variant there, it breaks the protein, just doesn't work anymore. But we know there are other diseases that are caused by the opposite, basically, where you put a missense variant in and suddenly it's doing something new that it shouldn't have ever done before. And predicting the different, developing a classifier that predicts both of those different cases and all the varieties in between has been a real challenge for the field and something that we haven't been able to solve yet by just throwing every disease variant.
I mean, I believe you can't do it today because you just don't have enough data.
I think even with them,
One thing to appreciate about the variants that we're talking about is that they're really, really rare. Like rare enough that if you sequenced everybody on the planet, you might have on the order of 10 instances of each variant. So getting the number of examples that you're talking about is actually not going to work. If you talk to genetics people and genomics people, 15 years ago, they thought exactly what you thought. We're going to sequence 100,000 people, or however many you need, 1 million. We're going to find all the variants that cause disease. There's a whole era of genomics that was born and has now basically died on that theory. And what's emerged is that everybody's genome and the variants therein are sufficiently rare that we would have to go far enough in the future that we've populated Mars and there are a couple 100 billion people alive, and then we could do the machine learning exercise. So I didn't know.
Got it.
Yeah, we don't have the resources even if we sequence the whole planet, basically. Yeah. So, like I've been hopefully saying, variant interpretation is a major challenge. What I didn't tell you in our merge work so far is that 3.5% had an actionable result, positive result, but an additional on top of that, 15% had a variant of uncertain significance in a gene that we know has actionable variants in. So these aren't even genes that we haven't identified yet. These are genes that have variants in them that we know, can initiate clinical action today. And 15% of people we sequence we have a variant we can't tell them it has from one or the other. And that's a little bit scary. Like we've just been discussing, computational interpretation methods haven't worked. Doug's right when he says it's been basically an era of human genetics over the past decade that thought that we could approach this with the amount of data we have at hand, and has since found out that we can't. Likewise, generating functional evidence for every possible variant out there is really tough. Historically, it's been hard to make all of these many variants at once. It's been hard to measure them all at once. And also, interpreting the functional evidence that we get back often requires engagement with patients and families, which is something that many of the people who are doing functional studies or the people doing computational evidence don't have access to. So there's a lot of power in joining large population cohorts that contain a variety of diseases up with patient groups for rare diseases and also computational folks working on this interpretation question. So what does Cadasil have to do with all this? So after I got my PhD in genome sciences, I moved over to medical genetics to work with Gail to get a bit more of experience thinking about the medical side of things. I'm a population geneticist, is my background, and only recently had decided that maybe medicine type stuff would be interesting. So I started going to Case Conference, which is where we have a great medical genetics clinic, and people like Sumi see patients, and once a week they gather and present their results to each other, and also to some interested scientists like me, they don't let their patients. And so it was at one of these Case conferences that Sumi happened to be presenting this disorder, Cadasil, and Notch3, the gene that caused it. I had never heard of this disorder, which I'm shocked to learn that, about it, how common it is relative to other rare diseases. And it really kind of, I had this light bulb moment that really was triggered by a few things that Sumi said and that I thought about. First was that, so the field of Cadasil research had recently been upended because this big EXAC database that I mentioned before, all these exomes came out. And the people had been doing Cadasil research on individual families with Cadasil, like we were talking about, and found all these mutations that they thought caused Cadasil, right? But then EXAC comes out. They don't have any Cadasil people. They're just people, unselected, random people. But it turns out all of these mutations, see, these are two different Cadasil cohorts. Frequency and the height of the bar is how many people had that mutation. And this is those same mutations in EXAC, again, people who aren't really supposed to have Cadasil. So there's overlap. There are people in this database who have these supposedly disease-causing mutations all across this entire gene. This is the layout of the whole gene. Again, these are the exons, the individual pieces of the gene. So they were left with this question in the field. What do we do? We're left with either two possibilities, right? Either these people in EXAC actually have Cadasil and it was undiagnosed or subclinical, or right? Or these variants that we thought actually caused Cadasil by identifying individual families don't. And there's a much more complicated story there, and we need to do a lot more. We need to learn a lot more about Cadasil before we can accurately classify these variants. So to me, that was really exciting because this is exactly the sort of problem that I'm interested in, is in applying large-scale genome sequencing to the problem of variant interpretation. But there was also another piece that kind of had hit off the light bulb moment, and that was Notch. So one great thing about being in genome sciences in general is that it's a department where there's people working on clinical human genetics right next to people working on flies and yeast and plants and all sorts of cool stuff. And so I got to see all kinds of really interesting cell biology and technology development in biology that was happening in genome sciences alongside the human genetics work that I was doing. And from that, I had heard about Notch before. It had rung a bell because it turns out, it's one thing Sumi didn't tell you is why it's called Notch. It's actually a bit of an interesting story. So Notch was first described in 1917 by Thomas Hunt Morgan, a really famous name in human genetics. He's basically the first geneticist. He was doing genetics before we even knew about the double helix, before we even knew about DNA, any of that. He has this really famous paper or book, depending on how you print it, called The Theory of the Gene, in which he describes all of the flies that he had in his pantry and all of their various different types of mutations and what they look like, and pedigrees of flies and how they were inherited. And so one of them happened to be this lady here. So these are the notches, where the notch comes from. Basically, in 1917, Thomas Hunt Morgan said, these guys have notches, we're going to call these flies Notch. And this gene, which has since become expanded out to Notch 1, Notch 2, Notch 3, Notch 4, a bunch of notches, turns out is one of the most important canonical signaling molecules in all of biology. It's a hub, and it touches nearly, especially in neuronal development and limb development, it really regulates how organisms develop and the pattern that cells grow in. So I thought, I knew this gene. I'd heard of Notch before. It's an old gene, and just a little trivia, it was the first ever dominant sex-linked recessive lethal that was ever mapped. So that's it's something we like to torture undergrad students with, particularly rare kind of genotype. But yeah. But it struck me as odd. There was this gene that was so canonical in biology that we've known about variation in since 1917, and we still can't sort all the variation in Notch, right? The ones that cause disease and the ones that don't. So I said, there's some smart people working on some really, really interesting stuff over in genome sciences in terms of how to make and measure these variants at high throughput. And so Notch might be a really perfect place where we can do this, and coupling some classic cell biology with some modern genetics and motivated patients and researchers to actually understand how we might sort this. So I really believe that our initiative to study Cadasil and Notch3 that we're now going to get into a bit more of the details of with Leah and Doug, it's going to not only advance our understanding of stroke and small vessel disease, but more importantly, I think, in general, it really provides a roadmap to interpret the variants across all of these genes, the ones that cause disease and the ones that don't. And one day when all of you guys do get your genomes back, because I believe it will happen in your lifetime, you'll have a little bit better understanding of what they do.
Everyone who's got a 23andMe account, raise your hand.
That's not a genome.
Well, it's something.
Yeah, it's a subset, yeah.
How tiny a subset is it?
Well, so that's the thing with 23andMe is they have picked individual spots of the genome that they know vary in people, and they just look at those spots. Whereas it's different than saying we're just going to read the whole book, right, from start to finish. Yeah.
If I want that, who should I send my spit to?
Actually, well, so I would recommend if you wanted the best direct-to-consumer right now, that one I was mentioning, Color. So theirs is actual sequencing.
Oops, right there.
On the right here.
Color Genomics.
Color Genomics. Yeah, theirs is actual sequencing, not the individual spots. But it's not your whole genome. It's a subset of the genome that we know is involved in cancer risk.
Nice.
But still, it is sequencing. So if you wanted to get sequenced, that's your best bet right now, which you can get pretty much off the shelf.
Well, that's not your entire exome. That's just.
No, yeah, I don't believe there's any exome. Yes, no.
Illumina will, though, right? Illumina will.
I think Illumina does DTC. I believe it do. Yeah, there are a few companies that will do one-off exomes for individuals if you want to, but I believe there's something like still 5 to 10 grand, depending on how much. Yeah. Yes.
Are there any predictive models out there that give you an idea on how possibly successful we can be on predicting with so many variables? We must be trying to look ahead and anticipate, because the more you get into this, the more sort of quasi-ethical questions you're going to get. How much do you want to know? And how certain are you that you may have something? All these sort of
this
perilous.
This is a major challenge, and something that I work on a lot in Emerge, in investigating and talking to people about their experience of getting these variants back. And luckily the field is very much, at least on the research end, moving towards opting in for results, for certain categories of results.
Right.
Except in the cases where we know that this could save your life or the lives of your family members, right? But for things that aren't like that say like, oh, maybe you just are slightly more tolerant of aspirin than someone else, for example. Those kind of things, people getting their genome sequenced, if they're doing it right and ethically, are either giving you access to everything or allowing you to pick and choose what you would like to receive back.
Good.
Yeah.
It's somewhat Cadasil naive question, but is it only Notch3 and no other Notch genes have pathogenic mutations for Cadasil?
As defined, it's just Notch3. There's diseases that are similar that have a different inheritance pattern, but Cadasil is just Notch3.
Any more other questions before I hand it over to Leah to talk a little bit about how we make these cells, or these variant cells? All right.
So I'm going to talk to you guys about the actual technology, how we make and test all the mutations in a gene. And Adam just kind of told you why we want to do that, right? So there's all of these different variants and all of these different genes, and we still don't know what they do. So one of the ways that we can test that is actually make them and do some cell biology or some assay to say, well, Are these cells still functional? Is this protein still functional or not? Okay. So I'm going to give a little bit of a molecular biology overview so we're all on the same page. I'm going to introduce you to our technology, talk about some data that we've generated in the lab over the last couple of years, and then our future plans for Notch 3. Okay. So I just want to make sure we're all on the same page because so the rest of the talk makes sense. Okay. So Adam told you that your genome is the DNA nucleotides. And then so each 3 of those encode 1 amino acid. And in this case, this sequence of amino acids, or this sequence of nucleotides, is spelling out in amino acids GRAY. So glycine, arginine, alanine, tyrosine. Okay? And so it's a triplet code. And I want to explain what I mean by variant or mutation. Okay? So right here, we're going to put in a variant. This is a single nucleotide variant, right? So we've changed one base that has now changed the code so that this is an E. Okay? And so we've gone from gray to gray. And if we use an English language analogy, we're very good at predicting that these 2 words mean the exact same thing. They're describing a color, right? All right. But what happens is if we change this now to a G, we've changed our amino acid to a glycine. This word is no longer a word, right? It's completely broken. And if we use the English language analogy, this protein is broken. Okay. The problem is that we absolutely are abysmal at telling the difference between, a variant that should still work and a mutation that's probably pathogenic and is going to break this underlying protein. Yes.
I should have asked this of Adam, but I was curious the difference between variation, mutation, and missense.
Yes, so these are words we throw around that actually do have a different meaning. So a variant is something that is just different between, a reference sequence, right? So or a reference sequence and somebody else.
Okay.
And it probably, if it doesn't cause disease, or if we don't know it causes disease, we call it a variant. So that could mean that somebody's got blue eyes and somebody's got brown eyes. It clearly has no health impact, right? So it's just a variant. If it does cause disease or causes a health impact, then it's a mutation.
And missense is?
Missense is one of these spelling mistakes, right? So if I actually change this G to any other base, there's degeneracy in the code. So this stays a glycine.
But if that got deleted, then all the other letters after it would shift down, and that would be a different type of...
That's a frame shift.
Yeah.
Yes. Right, exactly. And then there's nonsense too, and we'll talk about that later. And that's mutations. So some of these triplet codes code for a stop. So like the protein just stops getting made, and so you get half a protein. And those we're pretty good at. Those we know are bad, generally.
Didn't talk about
Yay, we can do that. So that's okay. So as I just said, we're bad at this. So this is why, this is kind of a lead into like why we need to do these assays. All right, so as Adam just told you, genetic databases are full of these variants where we have no idea what the variant does to the protein, so therefore we have no idea what the variant has to do with health, right? So this is our biggest genetics database, ClinVar, just in accounting, and everything in gray is either a variant of uncertain significance or these worse things, which are conflicting reports, meaning one company is telling patients one thing, like maybe it's benign, and another company is saying something else. So that's a pretty terrible category. And then,
Well, research papers do that all the time. So you can't just blame the companies, right?
And you
The
should.
repeatability problem in science is a big issue.
The major issue here is that often if it's in the US, it won't get reported or considered by the doctor, or the opposite, over-considered.
Right. Right, and I'll give you an example with breast cancer in a minute. So, Notch 3 variants. We want to know exactly which ones cause Cadasil, maybe which ones might have subclinical phenotypes, which ones don't cause Cadasil, and what they do to the protein. Because the more we can learn about the protein's biochemistry and its biophysical properties, the more we can learn about how the disease works. Okay. So Notch3. So this is currently my lab. This little rack in here has a whole bunch of Notch3 constructs that I got from the lab at Harvard. It's the only thing in the fridge. We're just about to start up. It turns out I work for a very large bureaucracy. So starting up has been a little slower than I care to talk about. So I'm going to tell you about work I've done before on a very famous protein. So you might be able to really understand why this is such a problem and why we would study BRCA1. So BRCA1 is a gene that, when mutated, can cause or is associated with up to an 80% lifetime risk of breast cancer. Okay, so this is, and then the good thing about BRCA1 is that if you know you have a pathogenic mutation, there are steps that can be taken to prevent breast cancer or ovarian cancer, but they're life-changing, right? So it's a double mastectomy, oophorectomy, immediate menopause, right? So these are not, you don't want to make a mistake with one of these variants, right? You want to know if it really causes disease or if it really doesn't. And the problem is with these variants of uncertain significance is that when these test results come back, these people know that they've got breast cancer in the family, and then they're told that they've got a variant of uncertain significance, so they've got to change, right? And even the doctors really don't know what to do with that. And people end up getting these pretty serious surgeries for variants of uncertain significance, which is not an awesome medical outcome, okay? And so it's confusing to patients, it's confusing to doctors, and this outcome of the test is not something that people really expect to happen, right? They expect an answer, not a non-answer, right? So this is BRCA1. This is, again, scraping that clinical database, and it turns out that there are some almost 2,000 variants in this protein in that database. It's a very big protein, and it's also been sequenced a lot, right? So the more you sequence, the more you find, which is what Doug was saying, that all of these are just so rare that the more you sequence, the more you find, and they're all very rare, so they all kind of end up falling into this category of variants of uncertain significance. And then
Those are just the ones you stumbled into.
Exactly, so there's a lot more out there.
Maybe.
Yes.
So if you weighted that by number of people who actually have the variant, what would that chart be?
So the chart actually, this would go way down because these are rare. So a lot of these are kind of unique instances. So a lot of the pathogenic variants are founder mutations. So, the first white people who moved to French Canada had a BRCA mutation. So there's a big population of those people with that specific mutation. So, there's a 5 or 6 founder mutations. There would be more BRCA. There's a bunch of founder mutations. So there's a Spanish mutation. So people of Spanish descent generally end up having that single variant. But yeah, but in all of the million of women that have been tested for this, they've found all of this rare variation. Okay, that's an excellent question, by the way. Okay, so going back to this gray analogy, if you take the wild type protein and say it has these four amino acids in a row, what we could do is actually make this protein and see if it folds correctly, see if it does its job in the cell. There's a whole bunch of different assays we can do. BRCA1 happens to be important for DNA repair, so we can do DNA repair assays, stuff like that. So what we would do to test this rare E variant is we would go and do the molecular biology to make the variant, and then we would take some other variant, say this G variant, and we would also make this variant in a single tube, and then what we would do is we would do some experiment, maybe it's looking at its ability to repair DNA, and then we would say, okay, well, this is the wild type one, this is the normal one, and that one works, great. This E variant, which we would expect to work, given our English language analogy, also works, but this one, which is all kind of, what is that? my husband, who's in here. That one doesn't work. So maybe that one is associated, might be associated with disease. Okay, but this is really slow. Okay, so BRCA1 was cloned 22 years ago, right? So if somebody was going to actually do all of those experiments, they would have been done, right? They're not easy. It's hard to do this. It takes a lot of time, and it's kind of pointless because every time, every month this genetics database updates. There's another 50 variants in BRCA1 that have been found. So it's kind of, you'd be running after this. Yeah.
And wouldn't that only work on soluble proteins? Not really very with the matrix proteins to develop assays for.
There's all sorts of ways. So this would be like a purified protein, but yeah, we can do cell-based assays for a lot of these too, so we can express them in the cells. Things, and I'll talk to you about how we think we're going to do some of the notch stuff. Okay, so we've developed, and Doug published the first paper on an assay like this, where what we did to twist this is we make all of the variants, all of the possible variants in the protein in one tube. We then put them into cells or something like that, and then we do the functional assay. So again, we can test for DNA repair. We can see if by putting these proteins into the cell, whether they survive or not. Okay, and the twist is that we use DNA sequencing. So instead of actually using a DNA sequencer to sequence a genome, we just sequence these little pieces of DNA and use it as a very expensive counting machine. Okay? And so we sequence all of these variants, and we can tell whether one specific variant is going down in the population. So maybe these red cells expressing this red variant are dying. So we can track by counting whether they're going away or they're rising, right? So then we can go back and say, okay, well, this variant is, we're losing it from the population. Maybe it's not functional. And in this case, we can actually look at thousands of these in one experiment, right? So we can start marching across genes like BRCA1 or Notch3 and look at every possible variant. So we're not, running against time to say, well, these are the variants that were in this database today. Well, what are they going to be tomorrow? So we're going to look at every possible variant.
So sorry, I don't understand.
Yeah.
So what's the cause of the population of variant N in your diagram here to decline?
So for BRCA1, I will give you an example. So BRCA1, if you lose it in a certain cell line that we have a cell line that is haploid, which means it only has one copy of its genome instead of two copies like it should. And if you disrupt BRCA1 or mutate it in any way, the cells just die. Right?
Okay.
So
they're especially sensitive to BRCA not working.
Yeah, so again, BRCA1 and 2 actually are DNA repair proteins. So you imagine if a cell only has one copy of its genome instead of two, it might be very sensitive to not having its DNA.
I see.
repair.
And so you're, even though the population is all mixed together for these different variants, you're able to still on the other end count this is variant 123
Exactly.
that was modified in that population, and that's what the sequencer is doing, is it's enabling you to differentiate?
Because I can read the sequence of this variant, so I can say it was in this population 420 times, it was only in this population 4 times.
Okay.
That's gone down, so I would quantify that this way. And then maybe these purple guys stay about the same, and they look, those are pretty good.
And what's allowed you to do this stuff in parallel with thousands of variants at once?
2 things, our ability to write DNA and our ability to read DNA.
You have the DNA printers and the sequencers.
Exactly.
Okay.
So we can, basically order these kind of with a little bit more complication from a company. So it used to be much more difficult to actually make all of these different changes. We actually don't even know, in some cases, which changes we're making because we're just trying to make all of them. And as long as we can count them on the sequencer, it doesn't really matter. And so the biggest thing was the sequencers because we can sequence billions of things, And so we can count billions of things, which is the most important part. Yeah.
So is that later part why you don't do it with, if it's sort of random mutation, is that why you have to do it with a sequencer and not like multiplex PCR or something?
Exactly. Exactly. Because you need to know, you need to know the sequence, not just count.
If the cells died, then how do you count them?
Well, you just infer that they're going away for a reason.
But if you make random mutations, you don't know what mutations they have.
So we have an input population, so it's important that you have a population.
You sequence first,
You
and
sequence
then you grow.
first, and then,
I see. Okay.
so you kind of watch over time
Got it.
that they disappear. Yep. Okay, so one of the ways we can do this is using CRISPR-Cas9. So you guys might have heard of this like new miraculous tool that we now have in our arsenal as geneticists. Okay, so CRISPR-Cas9, so Cas9 is a protein, and it is a double-strand nuclease, meaning it cuts target DNA on both of the strands, right? And the way that the reason it does that is because CRISPR-Cas9 comes from bacteria, and what it's trying to do is protect itself from incoming DNA, so bacteria, viruses, phage. So it's trying to protect itself and protect its genome from being invaded by foreign DNA. Okay, so we, as a field, have repurposed this, and why it's so awesome is that it's this RNA-guided cutting machine, right? So all you have to do is load an RNA onto this Cas9, and we do this in a multitude of very easy ways. And the DNA sequence that can base pair to that RNA gets cut. Okay, we can cut the genome almost anywhere we want now, which is pretty awesome. Okay, so as I just said, we've got BRCA1 in these cells, and we decided to cut it in one of its protein coding sequence. And we didn't give these cells anything to repair the DNA with. And so what happens is when you cut DNA and it can't repair itself very well, you end up with one of those frame-shifting mutations, right? Because a couple of bases get chewed off the end, the two pieces of DNA come back together, and you've got a broken protein. And when you break BRCA1, the cells just die. When you break another protein that the cells don't need, they live. Okay? And we can take advantage of this to study what any possible mutation in BRCA1 does, because the ones that damage the protein will die off and they'll leave the population. Okay? So what we do then, so we're trying to make all of these variants. We take some exon, we target it with Cas9, and then we give it something to repair the DNA with. And what we give it to repair the DNA with, they have single nucleotide variants. Okay, so as it's repairing its DNA, one copy or one version of or one mutation ends up back in the genome of these cells. Okay, so then we can see after we do this editing, so that's called DNA editing. After we do this editing, we sequence the cells at day 5, and so we know if we've made all of those mutations or not. And then we watch until day 11. And at day 11, we break the cells open and we sequence that. We sequence across where we've made those edits again, and we count how many there were on day 5 and how many there were on day 11, and we can quantify the change. Okay. So to do that, so basically it's just a ratio of the day 11 over the day 5. That's it. Super easy. And so what we get this nice histogram of scores, and write it 0 are all the functional guys, and write it -2 are the non-functional, right? So we can separate this. We can separate the different variants out on the x-axis, and this is just the number of them. So these are those stop codons that I told you about. So this is what happens when you make half a protein. Nothing good anyway. And you can see that those are all non-functional. So that was a relief for us when we're building this assay. We're like, okay, it kind of works. And then synonymous. So I was telling you guys too that you can make DNA changes, but they don't change the protein. And so those are all functional. So another win. Okay. But the ones that are really hard are the missense ones, where they change the amino acid, they change the spelling of the protein, and we're really crappy at guessing which ones those are. And so we get a nice bimodal distribution of those. So those are in green here. And you get a nice bimodal distribution, and we can start to tell whether or not they're functional or non-functional by whether they overlap these synonymous changes or they overlap these stop codons. So we can put lines up, so whether they're functional or non-functional by whether or not they look like a stop codon or they look like a synonym, right? And what we really wanted to know was, well, for the variants that are known to be pathogenic or they're known to be benign, so there's red for pathogenic and blue for benign, how did we do? Like, this is a crazy cell line. Why would BRCA1 variants behave in this cell line like they would in breast or ovarian cancer cells, right? And as it turns out is that we get almost every single one of them right. Okay, so we're got very good specificity and sensitivity for being able to pick apart which variants might be disease causing and which ones aren't. Okay. The ones we get wrong, we actually think the database is wrong, to be quite honest with you. So most importantly though, here in yellow and green are those variants of uncertain significance and the conflicting reports, and we're able to really stratify those pretty nicely into functional and non-functional groups. So maybe, eventually when we figure out how to really use these scores in the clinic, we can say, okay, well, this person's VUS is probably functional, so we're going to say that that's a benign variant. But most importantly, so these are just the ones that are in the database now. We have all of them. So all of the regions of the protein that we targeted, we have an extra 3,000 different mutations or variants. So that if they were ever seen going forward in clinical BRCA testing, we can say, well, we already tested that one. We know it's pathogenic, we know it's benign, or actually somewhere in between.
And you're testing these in a petri dish or something?
Yeah, so it's a cell line that grows on plastic.
So you're not in the whole organism. So it's possible that what you thought was benign because the cells didn't die in an organism where there's interaction between cells, right?
That is absolutely true. But for all the ones where we know
I'm not
that
trying
it's
to
benign
distract
or
you.
pathogenic,
I'm just asking a question if that's
yes,
true.
no, that's absolutely true. But yeah, so
you have to use the tools you have.
Exactly.
I get it.
Yeah, it's hard to make 4,000 mice. The IO would not go for that.
Yeah.
Can you comment on the synonymous and non-functional?
Yes, that's these guys.
Yeah.
So synonymous changes are the ones where you can change a DNA base, but it doesn't change the protein, right? Because there's degeneracy in the genetic code, right? So GGG and GGC both make glycine, right? So that's where we change the DNA, but not the. So they shouldn't affect the protein because you're not changing the amino acids. It does kind of affect splicing, so that's why you get a very small percent that are actually non-functional. But we're not going to talk about those yet. Okay, so Notch 3. So how do we apply this to Notch 3? So as Sumi and Adam just told you, Notch 3 is this transmembrane protein. It's expressed as one whole protein, but then it gets cleaved and it interacts with itself at its N-terminus. And then it gets inserted into the cell membrane where it interacts with other cells expressing other proteins, right? This is
And then Notch 3 is being produced where?
in this cell.
So how does it get outside the cell?
It's exported through the Golgi endoplasmic reticulum. Yeah, so it's actually made, cleaved, put back together, inserted into the membrane. There's a lot of cell biology that goes along with Notch 3. Okay, then this little orange piece, which is a transcription factor, when it's activated, translocates the nucleus. It turns a whole bunch of genes on, which for Notch 3 says, make endothelium, or something of that nature. Okay. So how could we do an assay for this? So what we could do is put a little hat on it. We can express what we call a protein tag. So it's a little piece of protein that they make really good antibodies to, and we can just stick it right on notch 3. When it gets inserted into the membrane, we can look for it using an antibody or something like that that has a fluorophore. So it would actually make this cell kind of glow green. And then maybe this notch 3 though has a mutation in it, and it gets stuck in one of those exporting pathways. And so therefore, this antibody wouldn't have anything to bind to. This cell would be green, this cell wouldn't. We can separate that using a machine called a fluorescent activated cell sorter, which is pretty awesome. So we can separate those and sequence all of the different notch versions in the non-fluorescing cell and all the notch versions in the fluorescing cell and say, okay, well, which ones made it to the membrane? Because that's question number 1. If it doesn't even make it to the membrane, it's broken.
And that assumes the hat doesn't interfere with that behavior.
Exactly. We have to make sure that that's the case. But there are also antibodies to notch 3, so I don't think we even need to put anything on it. Okay. And then another thing that I told you notch 3 has to do is this little purple piece has to get in, or this little orange piece has to get into the nucleus to turn some genes on, right? So what we can do is we can actually take another protein that's a different transcription factor and fuse it onto this end of notch 3, okay? And then when that gets into the nucleus, this little piece of protein will turn on green fluorescent protein or something like that, turn these cells actually green. And again, we can separate the cells that have a good notch 3 from the ones that have a crappy ability or have lost the ability to get into the nucleus by how green these cells are. Right, so again, we can use those fluorescence activated cell sorters to say these are super green, these are not green at all. And again, we can sequence which notch was being expressed in those cells.
So I'm judging from your verbs tense that these are the ideas about how you would do this, but you haven't done these yet.
Yes.
Got it.
Yes, because
No worries.
Not for notch 3 yet.
not for notch 3.
For other proteins.
It does work for other proteins.
Right.
Yeah, so we're excited to get started with this. We have a bunch of the reagents, and we just need to really start getting going. So thank you, and I'm part of the Brotman-Beatty Institute for Precision Medicine, and we'll be doing a lot of this work.
Cool.
Is that an official name, Bat Lab?
Yes, the Brotman-Beatty Advanced Technology Lab. So yes, we kind of reverse engineered that. All right, any questions? Is that clear enough? Okay.
Okay, so I won't take up too much time because we're running a little bit late. But wanted to tell you a little bit about how we're using some of the data, like data that Leah has generated, this large-scale functional data, right? So measuring the functional consequences of every possible change in a gene. And so I'll tell you a little bit about some of the challenges and opportunities that arise there, and then end with kind of giving you our vision. And us here is sort of the group of individuals in genome sciences and in UW that have pioneered these technologies and have really turned UW into a world leader for this kind of science. Okay, so just to bring back a slide that Leah showed you, right? This is our distribution of variant effects for her really beautiful BRCA1 data, and what she told you about and what you can appreciate is that there's a very clean separation here, right, between pathogenic variants and benign variants. And so when we're thinking about like using this data to make a predictive model of pathogenicity, the model is really, really simple, right? The model, as Leah said, is like you draw some lines and you have a classifier. You have any variant that ends up with a functional score on this side of the line, we think that one's pathogenic, and vice versa for benign. So this is an example where there's a very clean relationship between a variant's functional effect and disease risk. This is what we hope for. And sometimes it works like this. But there are some complications that are worth thinking about in this paradigm, of using functional data to predict pathogenicity. One of those complications, so I'll just tell you about one of those complications and sort of how we're thinking about it, both in terms of technological solutions and computational solutions. So one complication can arise when the gene that we're interested in is related to more than one disease, right? So variants in a gene cause multiple different diseases. And then understanding how a particular variant might confer a risk to one disease or another becomes quite complicated if you only have if you only measure like a single functional effect, right? Like say how cells grow when you introduce a variant. And so actually, Notch3 is one such gene. There are a couple of diseases besides Cadasil, although much more rare, that are in the literature that seem to be associated with variants of Notch3. And so this gets back to the point that Sumi made and was made again by Leah. Understanding the functional nature of each of those variants is key to understanding and allowing us to predict whether a particular variant is going to cause Cadasil or say one of the other diseases that adheres to Notch3. So our solution for this is to build technologies that layer on to the kinds of things that Leah was talking about, right? So she told you how we can make big libraries of variants in cells, and she told you about a cell growth-based assay, right, where we can measure how variants of BRCA1, say, alter cell growth. She also told you about some of our ideas for analyzing Notch3, measuring Notch3's activity as a transcription factor, and also its ability to be trafficked to the surface. So my lab is working on developing microscope-based technologies that take advantage of computer vision, automated image analysis, to basically analyze millions of cells expressing different variants for changes in cell morphology, so like cell size, shape, and also changes in cell behavior, right? So how far cells move, how fast they move, and how they're internally organized. And the hypothesis that's driving this work is that when we collect for each variant a large number of different types of data that tell us what that variant does, then we'll be able to make better inferences about how that variant relates to disease. Which this sort of gets to your question. We're stuck as experimental biologists because we can't do experiments on people, at least not ethically. What we can do are multiplex experiments on cells. And our idea is that the more pieces of data we gather for each variant, the better our inferences about variant pathogenicity will be. So that's sort of how we solve the problem in the lab. And
you're not really reducing cycle time, you're just doing it in parallel. That's how you're getting more.
Correct. That's exactly right. And we never can reduce cycle time because these guys take 24 hours to divide, and that's that. And actually, Leo was talking about mice. Like mice that's a 2-year experiment, right? At least with cells, we got a couple weeks. That's another big advantage. Okay, so this is just the point that if you've measured many different features of a variant's effect, then you can combine all of those data sets to make inferences about variant pathogenicity. And that's the sort of data science side of things, right? So using machine learning to combine all of the data that we generate with what we know, what the ground truth is, those variants of known effect that Leah was talking about, to develop predictions about pathogenicity that are rooted in the data that we've collected. So that's kind of one of the complications of what we're attempting to do and how we're sort of addressing that complication. Okay, so I want to end by telling you just a little bit about our answer to a question. So what can we do if we actually don't have functional data yet? This is just a slide showing you the structure of many different proteins. And it turns out there are about 4,000 disease-related genes in the human genome, each of which includes a protein, right? As we've articulated, our vision is to collect functional data for every possible variant in each one of those genes. Right now, we're at about 30 out of 4,000, right? So we've established a beachhead, we've demonstrated that our technology works, and we're working to scale it up. Notch3, of course, is one of the genes that we hope to add to this list next. But there are still a whole lot of disease-related proteins that we don't have functional data for. So going back to a point that Adam made, one thing we can do is develop a computational predictor of a variance effect using data that we do have. So we don't have functional data, but we have other types of data, like evolutionary data, protein structure data. We use that to make a prediction. So we've been working on developing a new and better computational variant effect predictor. And the secret sauce here is some of the functional data that I told you about, right? So, well, actually, Leah told you about it. So this is just a slide of a bunch of different data sets that we and others generated. And each one of these lines here is a different protein. And this is just some statistics about the mutational data set that we're in possession of. And I want you to pay attention here to the data set completeness, right? So for each of these proteins, we've measured all, or almost all, of the possible mutations, right? We've measured the effect of all or almost all of the possible mutations. And this is what's really special about the data we have, and it's what will allow us ultimately to train a better variant effect predictor, computational variant effect predictor. And I want to illustrate that with just a little analogy or example. So let's say I gave you the task of developing a computational model of how buildings work. Okay, so how rooms and plumbing and elevators come together to make a functional building. And then I offered you two different types of data to train your model. Okay, so one type, in both cases, you're going to get 20,000 drawings of rooms, okay, from buildings in the Seattle area, right? And in one case, you're going to get 20,000 rooms selected at random. So you'll get like my bathroom and your office and maybe your closet. I don't know. Each of the buildings in the city will contribute 1 or 0 rooms, and that's one type of data set. Another type of data set would be you get 20,000 rooms drawn from a very small set of buildings, right? So you get the complete plans for the whole building. And I hope you can see that if you're trying to build a model that tells you how a building works, this type of data is going to be way more helpful. And so computational variant effect predictors that were trained before the era of the kind of large-scale data that we produce, we're using this kind of data, very sparse, 1 or 2 mutations per gene across the whole genome. Our data is more like this, where we have the effects of all the mutations in a much admittedly smaller set of genes. So we used a decision tree-based learning algorithm to train the model that we call Envision, and I'm certain, don't worry, now we have 30 more slides of how it works. Sorry, one slide. This is the predictions of our model versus measured protein functional scores. And I just want you to appreciate that the correlation is quite good. There are many ways to assess the performance of this type of model, and if you have questions, you can answer them. But our model does a fair bit better than other models, and it's also quantitative. That's the other big innovation, right? So you see that we're on a continuous scale here, right? Rather than trying to classify as like good or bad. So those are sort of 2 innovations, but I want to be sort of real with you and end on the fact that like even though this is better, it is still absolutely not going to pass Adam's sniff test, right? We are not going to go into the clinic and say, okay, it's time to have a mastectomy or not based on these predictions. So they help to guide us. They're better than what we had before, but at the end of the day, our view. There's a screen. Gratuitous screenshot of the browser. If you're a science lab and you develop a piece of software good enough to like put on the web, you're very proud. Because we're not software engineers. So our vision, though, because we know that predictions are not going to be enough on their own, is to make and test the effect of every variant in every disease-related gene in the human genome, and to do that in some number of years that are less than what it will take for us to retire. I think that we're, who knows what, a decade away maybe from having that goal accomplished. Maybe 15 years, I don't know. But that's what it's going to take, because as has come out of the whole discussion that we've had tonight, right, most of the variants that we find in the human genome are too rare to know about by just going out there and sequencing more people. And we can never get anywhere with one-at-a-time functional testing, right? So waiting for a variant to show up. So we think that our proposed solution, which is testing en masse, is really the only way to go, and that's what we're championing, and that's what I have to say. So I can take questions. I don't know if there's a discussion plan now or.
So the 30 number you gave earlier is for your Envision for this testing of actual testing the variant genes, the 44 of the 4,000.
Oh, yeah, that's sort of where we are on the progress bar. But
that's 30 is your number for your group.
No, 30 is sort of for the whole enterprise. But we are
But just in UW, not the world.
No, that's the world.
Okay.
Of which we're the majority, like 28 out of 30.
30 out of 40.
So this question might be for any of the four speakers, but let's say you get a test for Cadasil, and let's say you get a nucleotide variant report back that says C2793-2A greater than symbol G, heterozygous. Then you get about a long paragraph of genetic gobbledygook with the lone conclusion saying this variant is classified as pathogenic. Is there anywhere for a patient to go, or an advisor who has a 35-year background in medical research, who spent the last 2 months Googling and trying to find out what this means, to go to get any idea of hope, or, do I have a variant that is pathogenic, or do we know something that the patient doesn't know? Where are we in all this?
So that's a great question. I'll give you my perspective, and that's a basic science perspective. And then it would be great to hear from Leah, who's kind of a little bit more sort of it goes me, Leah, Adam, Sumi, from basic to clinic, I think. And so my perspective is that if it were me, I would go to a medical geneticist. I think in my view, those are the only people who actually hold the knowledge that you would need to get anywhere beyond that report today. And that's not a great place to be in. Medical geneticists are great, but there aren't that many of them. Leah and I are working on building a database that will attempt to address that question, at least for people who are not medical geneticists, but who have enough knowledge to maybe interpret some of the information that might be useful. So we're working on the problem, but I think right now we're in the realm of specialists, and that's my perspective.
In this test result, was there only one lab in the entire world that does this test, or are there other labs that do this test?
There are many labs, different labs that do it.
All right, it's your life, but don't trust one lab.
Well,
right?
Well, I don't know.
This is such a good.
Is that true?
It's been done.
It's real, right? So here's the thing. So that report saying that variant is pathogenic means that after someone sequenced your gene, or however much of your genome was on the test, a few different experts at whatever the testing company was sat down and looked at the gene and basically subjected any variant they saw to that whole classification rule system, that big old table of rules that I had. They said, okay, we can assign this rule to that variant, this rule, this rule, this rule. And they add up all the points, and they get to pathogenic, right? So they've gone through a process, and in your case, for Cadasil, most of the pathogenic ones are known. It's 3 or 4 that come up in a lot of different people. So that pathogenic result means that we're pretty sure that the variant you have causes disease. In terms of what to do about it, that's where it comes
I'm not, it's not so much a question of what to do about it versus sort of a prognosis. I think is a key factor for people that have been told that, and to know what to expect.
And that's why clearly I have a bias, but that's why I completely agree with probably all 4 people here that probably the best thing would be to go to a geneticist. Okay. And that's because I think a number of things, just to talk about from the science up. The report you get is the experts in the lab that are reviewing the variants and deciding is it likely pathogenic or not. And while Cadasil, we know a lot about what is pathogenic. Remember that's in 2018 or 2017.
Right.
And these annotations change with time. So that's one thing. And also it's important, this sort of gets to what you're saying, is that if there is a variant that is pathogenic or not pathogenic, but one of the things that I think, another one of the things that Adam was sort of interested in is that there is variability in the disease severity and prognosis, et cetera. And those are things that don't come out in your binary result. So, but a medical geneticist, when you come to our clinic,
But would that be based on the actual data in the nucleotide variant?
Well, not yet, but one thing we envision actually, so a step past this machine learning right here is not only yes, no, does the variant cause disease, But for these diseases that can present in a variety of ways, how will your specific genotype predict your clinical course through that disease?
Because I'm wondering, is there data amassed yet that correlates the variant with outcome?
I will tell you, so that the more common the disease is, the better our information is,
Right.
because we've just had more opportunities to see it. So
And the more common the variant.
Yes, and even more common the variant. So again, in BRCA, these founding mutations that we see a lot, we have a better understanding of the prognosis than we might variant we've only seen once.
So you wouldn't know until you talked about it.
No, but again, this is the power, what I think is unique about with Notch versus some of the other genes and experiments that Lee and Doug have talked about, is with a motivated group of patients and family members who are interested, following people with actual Cadasil mutations who are interested in getting their family members tested, it adds another feature essentially to Doug's model to get to this point. We also think that for Cadasil in particular, there's imaging data, right? So MRI is is always going to be collected, and so to use some of these maybe imaging processing techniques that are also connected back to the actual genotype will help too.
Great.
I think we're going to mill about probably if people want.
Yeah, you guys have been so efficient.
Yeah.
PowerPoint us all at the end of the day.
Information.
With line, I mean, I'm shocked.
Thank you all so much for coming.
Thank you.
Thank you.